we have already discussed the metabolism, the synthesis, and everything about the purine and pyrimidine synthesis. Purines and pyrimidines. Now we are going to synthesis. So, do you know pyrimidine and purine synthesis? Various immunosuppressive, antineoplastic, and antibiotic drugs interfere with the nucleotide synthesis. First, we will have the basics of why we need to interfere with anything that is producing DNA. So, problem with neoplastic cells is they proliferate abruptly, anonymously, without our control of apoptosis or anything. So, to control this proliferation, either we need to decrease the supply of the supplements which make up the cell division. What is the main ingredient in cell division is DNA. So, the DNA synthesis from your purines and pyrimidines. So, if we stop the synthesis of purines and pyrimidines, then obviously the cell proliferation will decrease. So it is using neoplasia treatment based production and purine based production or reuse from salvage pathway. What do you mean by salvage pathway? You already discussed. Once you have purines and pyrimidines, while you are degrading, you need again synthesis of purines and pyrimidines so that your nucleosides and nucleosides are resynthesized from their metabolites respectively. In pyrimidine based production, first we will discuss about pyrimidine based production. We know pyrimidine based production requires aspartate. We have already discussed aspartate, glutamine, glycine. Wait, I will show you. Glycine, aspartate, glutamine are required for this GAG. We have remember this as GAG. So for synthesis of pyrimidine, we need glutamine. So once the glutamine is combined with the carbon dioxide, you use 2 molecules of ATP in the presence of CPS2. What do you mean by CPS2? Carbonyl phosphate synthesis 2. There is another one, carbonyl phosphate synthase 1. The main difference between these two is carbonyl phosphate synthase 2 is used in DNA production, whereas your CPS1 is used in the metabolism of urea, which are basically your proteins. See, there is a mnemonic given over here. CPS1, one is present in mitochondria, urocyte found in liver. Cytosol, CPS2, cytosol, which is used in pyrimidine synthesis and it is found in most cells. Why it is found in most cells? Because you need DNA in every cell. And CPS1 is found in liver because obviously it undergoes metabolism. So once the carbonyl phosphate is formed, it combines with aspartate to form orotic acid. This orotic acid is converted into EMP, UDP, DUDP, DUMP, DTMP. And the main thing you have to remember is dihydrofolate reductase is used here. In pyrimidine synthesis, leflumine, fluorosil. How to remember? Anything that contains fly is pyrimid. Fly on pyrimid, fly or pyrimid, fluor pyrimid. Anything can be remembered. Leflumide inhibit dihydrofolate reductase dehydrogenase. Difluorouracil and its product capecitabine form 5-FDUMP, which inhibits thymidine synthase. So that's about the pyrimidine metabolism, purine metabolism. In purine metabolism, the phosphorybosal pyrophosphate synthase is formed, which is converted into IMP, which is your mercaptopurine and its prodrug azathioprine inhibit de novo purine synthesis. What do you mean by de novo purine synthesis? New synthesis, not from the salvage pathway. We use the raw ingredients and we make up the purines again. So this is inhibited. Azathioprine is metabolized by purine degradation pathway and can lead to immunosuppression when administered with xanthine oxidase inhibitor. Azathioprine is a precursor of 6-mercaptopurine. It inhibits lymphocyte proliferation by blocking nucleotide synthesis. Which nucleotide synthesis? Purines.
Metabolite of
It is used in
which is
rheumatoid arthritis, Crohn's disease,
your
glomerulonephritis, autoimmune conditions. It can lead to pancytopenia. So this is what we are searching for. 6-mercaptopurine is decreased by xanthine oxidase. Derivative of 6-mercaptopurine is a derivative of azathioprine. So if we give xanthine oxidase, this azathioprine is not metabolized, which leads to toxicity of
water xanthine oxidase inhibitors.
Water xanthine oxidase inhibitors, L-purine. So yeah, that's what we have
Azathioprine. Now
completed
we will go to mycophenolate mofetil and
azathioprine. Now we will go to
ribavirin.
mycophenolate
This
mofetil
inhibit
and
ionosyl
rib-
monophosphate.
ribavirin.
See,
This inhibit ionosyl monophosphate. See,
monophosphate
ionosyl monophosphate.
dehydrogenase. Both some
Monophosphate
can inhibit
dehydrogenase.
both purines and pyridines.
Both
This
some
include
can inhibit
hydroxyurea,
both purines and
methotrexate,
pyridines. These include
trimethoprim,
hydroxyurea,
pyrimethamine.
methotrexate, trimethoprim,
Hydroxyurea inhibits your
pyrimethamine.
ribonucleic reductase,
Hydroxyurea
whereas your
inhibits your
methotrexate,
ribonucleic
trimethoprim,
reductase, whereas your
pyrimethamine.
methotrexate,
They
trimethoprim,
inhibit dihydrofolate reductase and decrease
pyrimethamine.
the
They inhibit
deoxythiamine
dihydrofolate reductase and
monophosphate
decrease the
in humans,
deoxythiamine
methotrexate in
monophosphate
bacteria,
in
trimethoprim,
humans,
protozoa, pyrimethamine.
methotrexate
All
in
these are
bacteria, trimethoprim, protozoa,
drugs.
pyrimethamine. All these are
Sulfamethoxyl is combined with
drugs. Sulfamethoxyl is
trimethoprim is the
combined with
combination used in antibiotics
trimethoprim. This is the
to kill the
combination used
bacteria.
in antibiotics
So yeah,
to kill the
completing all
bacteria.
these purines pyrimidines.
So yeah,
Now we'll
completing all
jump
these
into the
purines,
genetic code.
pyrimidines, now
So there are
we jump into the genetic code.
any
So there are
DNA to
any
act as it's
DNA to
to
act as
transcribe into
it's
mRNA and to
to
form your
transcribe
protein.
into
There must be a
mRNA and to
specific code.
form your
What do you mean by
protein.
code?
There must be
Your
specific
code
code. What do
contains
you mean by
three
code?
amino acids.
Your code contains
Hmm.
three amino acids. Yeah,
Yeah,
your
your
genetic
genetic
code
code
contains
contains
three
three
amino
amino
acids.
acids.
These
These
three
three
amino
amino
acids
acids
carry
carry
specific
specific
sequence
sequence
such
such
that
that
they
they
encode
encode
to
to
only
only
one
one
type
type
of
of
amino
amino
acid.
acid.
You understand?
Understand?
Yeah.
Yeah.
So
So
we will
we will
talk
talk
here.
here.
See,
See,
genetic code. Genetic
genetic
code
code
features.
features.
Genetic
Genetic
code
code
is
is
unambiguous.
unambiguous.
Each
Each
codon
codon
specifies
specifies only
one
one
codon,
codon,
one
one
amino
amino
acid.
acid.
But
But
one
one
amino
amino
acid
acid
can be
can be
coded
coded
by
by
many
many
codons.
codons.
Have
Have
you
you
understand
understand
the
the
relationship?
relationship?
One
One
codon
codon
can
can
only
only
be
be
give
give
rise
rise
to
to
one
one
amino
amino
acid,
acid,
whereas
whereas
this
this
amino
amino
acid
acid
can
can
be
be
given
given
by
by
many
many
codons.
codons.
Yeah,
Yeah,
degenerative and
degenerative and
redundant,
redundant,
which
which
means
means
most
most
amino
amino
acids are
acids are
coded by
coded by
multiple
multiple
codons.
codons.
See,
See,
that's
that's
what
what
I
I
told
told
already.
already.
Wobbles
Wobbles
hypothesis.
hypothesis.
First
First
two
two
nucleotides
nucleotides
of
of
a codon
codon
are
are
essential
essential
for
for
anticodon
anticodon
recognition,
recognition,
while
while
the
the
third
third
nucleotide
nucleotide
can
can
differ.
differ.
Exceptions
Exceptions
methionine,
methionine,
AUG,
AUG,
and
and
tryptophan,
tryptophan,
UGG are
UGG are
encoded by
encoded by
only
only
one
one
codon.
codon. What
What do
do
you
you
mean by
mean by
wobble
wobble
hypothesis
hypothesis
is
is
the
the
first
first
two
two
in a
in a
codon
codon
there are
there are
three
three
nucleotides,
nucleotides,
right?
right?
Let's
Let's
say
say
it
it
A,
AG.
see
See
AG.
AG.
This
This
can
can
the
the
third
third
amino
amino
acids
acids
can
can
change
change
accordingly.
accordingly.
It
It
can
can
change
change
it
it
to
to
AU,
AU,
it
it
can
can
change
change
it
it
to
to
AEC.
AEC. But first two nucleotides
First
are essential for
nucleotides are
formation
essential
of
for
your amino
formation of
acid.
your
This is
amino
what
acid. This is what
Wobbles
Wobbles
hypothesis. It's
hypothesis.
commonless and
It's commonless and
non-overlapping, read from fixed
non-overlapping. Read
starting
from
point as
fixed
continuous
starting point as
sequence of
continuous
bits.
sequence of bits.
Exceptions are unwavering.
Exceptions are
It is
unwavering.
universal.
It is
Genetic codon
universal.
is
Genetic
conserved
codon is
throughout the
conserved
evolution,
throughout the
except
evolution,
in
except
this.
in
This is
this.
important.
This is important.
What we learned
What
another
we
thing
learned
about
another thing about
mitochondria does not have
mitochondria does not have
histones
histones
on the first
on the
page,
first page, and also it
contains
is
circular
contains circular
DNA.
DNA.
These both are
These both
important.
are
Yeah.
important. Yeah.
Coming to DNA
Coming to
replication.
DNA
DNA
replication.
replication occurs in
DNA
5'
replication
3'
occurs in 5'
direction.
3'
It is
direction.
mnemonic by
It is
mnemonic
mnemonic by
synthesis in a
mnemonic
continuous and
synthesis in a
discontinuous
continuous and
Okazaki
discontinuous
fragments.
Okazaki
What are these
fragments.
Okazaki
What are
fragments?
these
These
Okazaki
Okazaki
fragments? These
fragments
Okazaki
contents of your
fragments
RNA
contents
primer
of your RNA primer
with small
with
segment of
small segment of
DNA
DNA
present on your
present
lagging
on your
strand.
lagging
Don't
strand.
be confused.
Don't be confused. We'll be discussing it shortly.
Lagging strand.
Lagging
And
strand.
important thing
And
to remember is
important
this
thing
is
to remember is this is synthesized by
DNA
DNA
polymerase 3
polymerase
in
3
case of
in case of
prokaryotes.
prokaryotes.
And this is present in
And
both
this is present in both
eukaryotes as well as your
eukaryotes as well as
prokaryotes.
your prokaryotes.
Or given by eukaryotes
Or
and
given by
prokaryotes,
eukaryotes and prokaryotes, we are eukaryotes. It
nuclear
contains a
membrane.
nuclear membrane.
It is semi-conservative
And a semi-conservative
conservative
conservative
process. What do you mean by
process. What do
semi-conservative
you mean by
process?
semi-conservative process?
This is the
This is the
mother DNA from where the
mother DNA
replication
from
starts.
where the
Once the
replication starts.
DNA
Once
gets
the
replicated,
DNA gets
each
replicated,
strand of it
each
will
strand of it
divide like
will
this,
divide like this,
and it will
and
synthesize a
it will synthesize a
The parent strand will
the
synthesize the
parent
daughter
strand will
strand.
synthesize a daughter strand.
So these are some
So these are
sequences.
some
First we'll go through
sequences.
all
First
these
we'll
names
go
and then
through all
I'll
these
explain
names and then I'll
the process
explain
of DNA
the process
replication.
of DNA
Once the
replication.
origin of
Once the
replication,
origin of replication, particular
consensus sequences in
consensus sequences
genome
in
where DNA
genome
replication
where
begins,
DNA
maybe
replication
single
begins, maybe
prokaryotes or
single
multiple
prokaryotes or
eukaryotes.
multiple
80
eukaryotes.
sequences are
80
found
rich
in
sequences are
promoters and
found in
origin
promoters
of
and
replications, which is
origin of
known as O
replications,
site.
which is known as
So
O
coming
site.
to origin of
So coming to
replication,
origin of replication,
to
to
to to start the
to
DNA
to start
replication,
the
you need a
DNA
point
replication, you
where
need a
to start
point
the
where
replication.
to start the
The
replication.
point where the
The
replication
point
starts
where
is
the
called
replication
as
starts is
origin of
called as
replication.
origin of
So
replication.
origin of
So
replication is a
origin of
point which is
replication is a
selected
point which is
at where
selected
the maximum
at where the
adenine and
maximum adenine
inosine and
and adenosine
thymine are
and
there. Why
thymine are
we need
there.
to select
Why
this?
why we
See, we
need to
already
select this? See,
discussed
we already discussed before
glue
only glue.
where guanine and
Where guanine
cytosine are
and
present, they are
cytosine are
triple bond.
present,
So
they
it
are
is a
triple
strong one.
bond.
Whereas
So it's
your
a
AT
strong
rich
one. Whereas
sequence
your
are double
AT rich
bonds.
sequence are double
So
bonds.
this is the
So
region where
this is
the
the region
bond
where the
breakage is much
bond
more
breakage
easier
is much more easier
rather than going for the
rather
strong
than going
sequences
for the
where
strong sequences
guanine and
where
cytosine are
guanine
present.
and cytosine are present.
Replication fork, Y-shaped
Replication
region
fork,
along the
Y-shaped
DNA
region
templates
along
where
the DNA
leading and lagging
templates where
strands are
leading and lagging
synthesized.
strands are
See,
synthesized.
this is the
See,
replication
this is
fork.
the
This
replication
is
fork.
known as
This
replication
is
fork,
known as
where
replication fork,
the
where
DNA
the
synthesis
DNA
starts.
synthesis starts.
So the basic
So the
explanation is
basic explanation is
just imagine a thread
just
which is
imagine a thread which
wounded
is
over the other like
wounded
this.
over the other like this.
To start the
To
replication, we
start the
need to open
replication, we
up this
need to
DNA. To
open up
open up
this
this
DNA.
DNA, we
To
need to
open up
select
this
the site.
DNA,
The
we need
site
to select the
selectivity.
site. The
So,
site
what
selectivity.
does the
So,
ORI
with
contains?
what
80
does the
sequence.
ORI
Why do
contains?
80
80
sequence?
sequence. Why do 80
Because it's a
sequence?
double bond and
Because
very
it's a
easy to
double bond
break
and
there.
very easy to break
Replication
there.
fork is once you
Replication
open
fork
this
is once
DNA,
you open this
the DNA
DNA,
will
the
becomes
DNA
like
will
this. See,
becomes
it is
like this.
given here.
See, it is given
This
here.
is the form which
This
the
is the
DNA
form which
acquires
the
once
DNA
you
acquires once you
remove these doubles.
remove these towels.
Once you open up the DNA.
Once you open up the DNA.
Helicase unwind the
Helicases.
DNA
Helicase
template, the
unwind the DNA
replication fork.
template, the replication
Helicase
fork.
halves the DNA,
Helicase half
defect
the
is known as
DNA,
Bloom
defect is known
syndrome.
as
It is
Bloom
important. The
syndrome.
Bloom
It is
syndrome, the
important.
helicase
Bloom
defect
syndrome,
is
the
there.
helicase defect is there.
What do you mean by unwinding?
What do
See,
you
this
mean
is
by
what we
unwinding? See,
discussed.
this is what we discussed.
Unwinding is breaking out
Unwinding is
of
breaking
the
out
bonds and
of the
opening up the
bonds and
DNA so
opening
to
up
as
the
to form
DNA,
the
so to as
replication
to
fork.
form the replication fork.
Single stranded binding proteins
Single
prevent
stranded binding
strands from
proteins
re-ending
prevent
and
strands from
degradation. See,
re-ending and degradation.
once you
See,
unwind these
once you
strands,
unwind these
they are even
strands,
close
they
to
are
each other.
even close to each other.
They have
Even
got
they have got
they are
away,
unwinded. They are
they
very
are
close to
unwinded. They are very
proximity to
close
each
to
other. So to
proximity to each other.
stabilize
So
this
to stabilize this
unwinding, we give
unwinding, we
this
give
single
this
stranded binding
single
proteins.
standard
The
binding
single
proteins.
stranded
The
binding
single
proteins will
standard
prevent
binding proteins will
re-annealing of the
prevent
strands.
re-annealing of the strands.
DNA topoisomerase,
DNA topophosphorases,
creates
topophosphorases
a single
creates a single
topoisomerase 1 or double
topoisomerase 1 or
topoisomerase
double
2 stranded
topoisomerase
break
2
in the helix
stranded break in
to
the
add or
helix
remove
to add or
supercoils
remove
as needed
supercoils
due to
as needed due to
unwinding or overwinding of
unwinding or
DNA.
overwinding of DNA.
Yeah, the single
Yeah,
stranded
the
breaks in
single standard
helixes are
breaks in
added. This
helixes are
topomerase,
added.
what
This
they do?
topomerase is
See,
what they
once
do.
this
See,
is your
once
DNA,
this is your
and
DNA
you are
and
unwinding
you're
it. So
unwinding it,
the
so
proximal
the
lens will
proximal
get
lens will
obviously
get
supercoiled.
obviously supercoiled.
To prevent this
To
formation, what do
prevent
you mean
this
by
formation,
supercoils?
what do you
See,
mean by supercoils? See,
this is the DNA,
this is the
normal
DNA,
DNA.
normal DNA.
You are unwinding the
You're
DNA. So
unwinding
once
the
you remember,
DNA. So once
once
you
you
remember,
remove these
once you
bonds and you
remove these
unwind
bonds
this,
and you unwind
the
this,
proximal
the
segments
proximal
will get
segments
closer to each
will
other.
get closer to each other.
Are you understanding? So
You are
this is
understanding?
where
So
you open and
this
this is
is
the
where you open
replication
and
fork,
this is the
and the
replication fork,
proximal segments
and the
getting
proximal segments getting
supercoiled. So
super
to
coil.
prevent this, we add
So to prevent
topoisomerase
this, we add
primase.
topoisomerase
Makes
primase,
RNA primer for
makes
DNA
RNA primer for DNA
phase 3 to initiate
phase 3
replication.
to initiate replication.
So before the
So
start of
before
replication,
the
you need
start
a
of
primer.
replication, you need a
This is the
primer.
sequence from
This
where
is the
your
sequence
DNA
from where
synthesis
your
starts.
DNA synthesis starts.
DNA polymerase 3,
DNA
1
polymerase
DNA
3,
ligase
1
telomerism.
DNA ligase
Telomerase don't
telomerism.
get
Telomerase don't
overwhelmed.
get
We will discuss it
overwhelmed. We
slowly.
will discuss it
DNA
slowly.
polymerase 3 is found
DNA
in
polymerase
prokaryotes
3 is
only
found in
along its
prokaryotes
leading
only
stand by
along its
adding
leading stand by
deoxynuclease
adding
to 3'
deoxynuclease to 3'
elongate its log
elongate its log
lagging stand until it reaches
Langston
a
until
primer
it
of
reaches a primer
preceding
of
fragment. DNA
preceding
polymerase
fragment,
3,
DNA
5'
polymerase
3'
3,
synthesis
5'
and
3'
proofreads.
synthesis, and proofreads.
Don't confuse with
Don't
proofreads. We'll
confuse with
discuss
proofreads.
proofreads
We'll
with 3'
discuss
5'
proofreads with 3'
exonuclease.
5'
Drugs blocking
exonuclease.
DNA
Drugs
replication
blocking
often
DNA
have a
replication often have a
modified 3'
modified 3'
hydroxyl and thereby preventing
hydroxyl length, thereby preventing addition of next nucleotide chain termination. DNA polymerase 1 prokaryotes only degrades the RNA primer and replaces with the DNA.
Same functions as DNA
Same
polymerase
functions
3
as
also
DNA
acts as
polymerase 3
RNA
also
primer with
acts as RNA
fragmentary
primer with 5 prime 3 prime.
Exonuclease,
DNA
for different
ligase.
by ligase, it
For different
just
by
joins
ligase,
the
it
hands.
just joins the hands.
Yeah, we will go into the
Yeah,
process.
we will go into the process.
Don't be confused.
Don't be
Catalyze is the
confused.
formation of
Catalyze is the
phosphodiesterase
formation
bond
of
within the
phosphodiesterase bond within the
standard double stranded DNA.
standard
Joins
double stranded
Okazaki
DNA.
fragments.
Joins Okazaki
Ligase means
fragments.
links the
Ligase
DNA.
means links the DNA.
Telomeres are present in
Telomeres
telomerase are present in
eukaryotes only. A reverse transcriptase. This is important. RNA dependent.
DNA polymerase that adds
DNA polymerase that adds DNA.
This sequence is important.
This sequence is important.
T2A1G3. That's how I
T2A1G3.
remembered.
That's how I
Two
remembered.
T's.
Two T's. See.
Two T's.
Two T's.
So T.
So T.
First alphabet is A.
First
A.
alphabet
So
is A.
G3.
Yeah. So G3.
To the 3' end of
To the
chromosome
3'
to
end of
avoid the loss of
chromosome to
genetic material
avoid
which
the
is
loss of genetic
duplication,
material, which is duplication, upregulated in
progenitor cells and often
progenitor cells and often cancer, downregulates in age in progeria. Telomerase, THE for greatness and glory. I'll explain this. Wait.
See,
See,
once you have your DNA,
you have selected your origin of replication of site, which is known as ovary site, and you have added your helicase, and it leads to formation of your replication fork.
See, this is the
replication fork. So now you see origin of replication, helicase, replication fork. All these have been up to we are having clarity. Now we need the formation of our DNA. See, the main point over here is DNA synthesis always occurs from
5' to 3'.
5' to 3'. So the new strand that is formed is always 5', 3'. But the complementary strand or parent strand from which this is forming is there are some rules. This is the DNA. 3', 5'.
There is another strand.
Opposing it, which is antiparallel. 5', 3'. So if the new DNA is formed as per the universal rule, it is formed easily. In 5', 3' direction, you have no worries. This is known as leading strand.
The opposite strand,
which is 5', 3', will form DNA in also 5', 3' direction. But it forms in
segments.
This is known as lagging strand.
Before going to the
formation of lagging strand, first we will discuss about the leading strand.
You
will have three types of polymerases in prokaryotes. Remember prokaryotes, DNA polymerase 1, 2, 3. 2 is for repair if any damage happens to the DNA. 3 is the main polymerase which starts replication.
Your DNA
polymerase 3 has 5', 3' synthesis activity.
See, we have discussed
this 5', 3' synthesis activity. It is universal.
And we also
have proofread from 3' to 5'.
So what do you mean by this
proofreading?
Once the
DNA is forming, let's say this is parent DNA and this is our daughter DNA.
Let's say there is a break
over here, or the sequence that has been synthesized is not the usual sequence, but an abnormal sequence has been synthesized over here by your daughter DNA.
The replication is happening
in 5', 3' direction.
Now what will happen is
your DNA polymerase
1
or 3. They both possess your synthesis happening in this direction, but the proofreading happening in this direction, which means the DNA polymerase 1 and 3 check whether the sequences are formed correct or not, and they'll repair if there is any sequence that has been mismatched or abnormally placed over here, or an abnormal nucleotide has been placed over here, and they'll repair that.
And there is an extra
activity for DNA polymerase 1 in the lagging strand. We'll discuss that, no problem.
And also once the
DNA polymerase 3 has this only 3', 5' proofreading, but DNA polymerase 1 also have 5', 3' proofreading. This is used in lagging strand. We will discuss it.
So
these are the basics.
Now if the
DNA is forming replication fork, you have I'll go in the diagram.
See, 3',
3
So
prime,
since this
5
happening
prime.
is
So
5
since this
prime
happening is 5 prime,
neatly without any
neatly
confusion,
without
you
any
have your,
confusion, you
see
have
this is
your,
the RNA
see
primer,
this is the RNA primer,
and you have your
and
DNA
you have
polymerase
your
3. See
DNA
this is
polymerase 3. See
synthesizing
this is synthesizing
without
it
any disturbance.
without any
The whole
disturbance.
confusion
The
comes
whole
with the
confusion comes with
lagging
the
stand, because
lagging stand,
you
because
have
you
different
have different
segments that are synthesized
segments that are
individually,
synthesized
and they are
individually,
packed,
and they are packed,
they are
they are
connected each
they're
other,
connected
which
each
forms
other,
the lagging
which
stand.
forms the lagging
See,
stand.
you have
See,
RNA
you
primers
have RNA primers,
and you have DNA
and you
polymerase
have
3.
DNA polymerase 3.
Let's say your DNA
Let's
polymerase
say
3
your
is
DNA
have
polymerase
synthesized
3
up to
have
here.
synthesized up to here.
Once the synthesis has
Once
been
the
done up
synthesis
to
has
here,
been
what
done
will
up to
happen
here,
is
what will happen is
your DNA polymerase
your
3,
DNA
once
polymerase 3, once it
it
encounters
encounters
your
your RNA
RNA
primer
primer
of
of the
the next
next
segment,
segment,
it
it
will
will
just
just
leave the place.
leave
So
the
once it
place.
leaves the
So
place, you
once it
need
leaves the place, you need
something to bind this
something to
segment of
bind this
DNA
segment
to
of
this next segment
DNA
of
to this
DNA.
next segment of
And
DNA.
while the
And
process
while the
of
process
uh,
of uh,
attaching one proximal
attaching
segment
one
of DNA
proximal segment
with the
of
distal
DNA
segment of
with the distal
DNA,
segment
you
of
need to
DNA, you need
see
to
you have RNA.
see
You
you
don't
have
need
RNA.
RNA in
You don't
between
need
your
RNA in
DNA.
between your
So you need to
DNA.
remove this
So you need to
DNA,
remove this
and you
DNA,
need to attach
and
this
you need to
DNA to
attach
each other,
this
so
DNA
that you
to
make a
each other so
straight
that you
segment
make a
of DNA.
straight segment of
So
DNA.
to
So
remove this primer,
to
we have
remove this primer, we
DNA
have DNA
polymerase 1.
polymerase 1.
This is the one which
This
comes
is
over
the
here
one which comes over
and
here
removes this
and
primer and
removes this
forms
primer
this
and
segment.
forms this
Once this
segment.
segment is
Once
formed,
the segment
DNA
is formed, DNA
ligase
ligase
attaches this both
attaches this
segments.
both segments.
Okay, so this is the
Okay,
process.
so this
Once
is
I'll
the
explain
process.
here
Once I'll
again.
explain here again.
Yeah,
Yeah,
this
see,
is your,
this is your.
please concentrate, this is
Please
important.
concentrate. This is important.
5 prime
5
3
prime,
prime.
3 prime.
You need synthesis
You
from
need synthesis from
5 prime to 3
5
prime
prime to
in
3 prime
daughter DNA.
in
But
daughter
this is
DNA.
the lagging
But this is
stand,
the
so
lagging stand.
it's
So
not happening
it's
as
not
easy
happening
as
as
we think.
easy as we think.
Before the formation of
Before
this,
the
we need
formation
RNA
of this,
primers
we need
attachment
RNA
over
primers attachment
different
over the
segments of your
different
parent
segments
DNA.
of your
Once
parent DNA.
this RNA,
Once
these are
these
RNA
RNA,
primers.
these are RNA primers.
Once these RNA primers
Once
are
these
formed,
RNA
then you
primers are
will get
formed,
your
then you will
DNA
get your
polymerase 3.
DNA polymerase 3.
Let's say your
Let's
DNA
say your
polymerase 3
DNA
has
polymerase
synthesized
3
some
has
segment of
synthesized
DNA.
some segment of DNA.
In each place, it has
In
DNA
each
polymerase
place, it
3.
has DNA polymerase 3.
Each place.
Each
Here,
place.
here, here,
Here,
everywhere
here,
we have
here,
DNA
everywhere
polymerase
we
3.
have DNA
It
polymerase.
synthesizes a small
It
segment of
synthesizes a
DNA,
small segment of
and
DNA,
this primer,
and this
along with
primer,
your
along with your
DNA segment that has been
DNA
initially
segment that
synthesized
has
by
been
DNA
initially
polymerase
synthesized
3,
by
this is
DNA
also known
polymerase.
as
This is
Okazaki
also known as
fragment.
Okazaki fragment.
Okay,
Okay,
clear? Yeah.
clear?
Up
Yeah.
to now, what we have,
Up
we
to
have
now, what
RNA
we
primer,
have,
we
we have
have DNA
RNA primer,
polymerase
we
3.
have DNA polymerase 3.
What are our
What
further
are
goals?
our further goals?
We need to remove
We
this
need
RNA
to
primer,
remove this
and we
RNA
need to
primer,
attach
and
this
we need to
DNA
attach this
segment with this
DNA
DNA
segment
segment.
with this DNA segment.
So now we
So
have
now we
DNA
have
polymerase
DNA
1.
polymerase
Next step.
1. Next step.
DNA polymerase 1
DNA
removes this
polymerase 1
RNA primer
removes this
and
RNA primer
synthesizes a
and
DNA.
synthesizes a DNA.
That's the one name
That's the one one
that
name
has been
that
done. The
has
next step
been
that is
done.
to
The
be done
next
is
step
we
that
need to
is to be done
ligate
is
this
we
the
need to ligate this the
proximal segment and the distal
proximal
segment.
segment and the
We
distal
have
segment.
over here
We have over here
DNA ligase.
DNA ligase.
So that's how your
So that's how
replication
your
works.
replication works.
Hope I have cleared
Hope
all
I
your
have cleared all your
confusions, and I am super
confusions
clear
and I
over
am
here.
super clear over here.
This topic up to
This
replication,
topic up
is
to
it okay?
replication. Is it okay?